class 12. Today we will be starting with the new chapter, that is molecular basis of inheritance. What is molecular basis of inheritance? In the previous chapter, we had studied about inheritance, which is by the variations. Okay. Now, how this variation is coming? These variation is coming by the genetics. And what is genetics? This genetics generally is comprising of the DNA, the RNA, nucleic acid. So we have to study all these topics in the molecular basis of inheritance, that is replication. Replication is what is multiplication. Transcription is there, where transcription, the DNA into mRNA. Translation, mRNA into protein is there. Transcription units are there. Cistron is there. Splicing is there. Splicing where the removing of the non-coding sequences are there. So many more things, and interesting DNA fingerprinting, regulation of gene expression we are going to study in this chapter. Molecular basis of inheritance, which involves the study of the genes, the nucleotides, the nucleosomes, genetic variation in the heredity. DNA and RNA and genetic code, which forms the basis of molecular basis of inheritance. They transmit the hereditary genes from the parent to the offspring. How it is. Now coming to the nucleic acid. There are two types of nitrogenous bases, that is purine, which is adenine and guanine, and pyrimidine, which is consisting of cytosine, uracil, and thymine. Uracil, which is present in the RNA, cytosine and thymine in the DNA. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. You can simply see the structure of the nucleic acid, the DNA, deoxyribonucleic acid, and the ribonucleic acid. You can just go through it as it is important. So a nucleotide which consists of nitrogenous base, pentose sugar, and phosphate group. Nitrogenous base, which consists of purine and pyrimidine. Pentose sugar, which is either can be ribose or deoxyribose. If ribose sugar, then ribonucleic acid. If deoxyribose sugar, then deoxyribonucleic acid with a phosphate group is present. A, which combines with the T, that is adenine with thymine, and cytosine with guanine. Adenine with thymine with the 2 hydrogen bond, and cytosine with guanine with the 3 hydrogen bond. DNA is an acidic substance which is present in the nucleus, and it was first discovered by Frederick Mischer in 1869, and he named it nuclein. This is the double-stranded polynucleotide chain of. What are the silent features of a DNA double helix? These are very important points. Now, the silent feature is that it is made up of two polynucleotide chains where the backbone considered by the sugar phosphate, which is projected inside the helix. That can be right-handed and that can be left-handed also. Now, these two polynucleotide chains have the antiparallel polarity. Antiparallel antiparallel means if one strand is 5 prime to 3 prime, then the other strand will be 3 prime to 5 prime. What do you mean by prime? Over here is the location which is vacant and where the new group will be attached, that is 5 prime means 5th position and the 3rd position. So they are opposite. Okay. The bases on the opposite strand are connected through the hydrogen bonds. Adenine forms 2 hydrogen bond with thymine, and guanine forms with the 3 hydrogen bond with cytosine with the opposite. Therefore, a purine always pairs with the pyrimidine and pyrimidine with the purine. The 2-strand coils in the right-handed fashion, and each turn of helix is 3.5 nanometer or 34 angstrom unit, which consisting of 10 nucleotides. This is very important data. These nucleotides are at a distance of 0.34 nanometer, 3.4 angstrom unit. The helix is stable because of the base pairs that stack over one another and the hydrogen bonds that holds the base together, which is very important. How is the packaging of DNA occurs? DNA is what? Negatively charged, acidic. So over here, how the pink portion in this figure, you can easily see the pink portion, the round coil is the DNA, and the big green ball over here, which consists of the histone proteins. Histone proteins will be basic in nature and they are in the form of an octamer called as histone octamer. Now, if you calculate the length of DNA in a typical mammalian cell, it is approximately is 2.2 meters. The dimensions of a typical nucleus is only about 10 to 6 meters. Then how does such a long polymer fits in the nucleus of the cell? Just think about how long it is and how it is inside the body. Prokaryotes like E. coli do not have a definite nucleus. Here, the negatively charged DNA is held together in a large loops by positively charged proteins in a structure called as nucleoid. So you will come to know over here difference between nucleoid and nucleolus. Because in eukaryotes, nucleolus is present where the histone protein is present. Now, the negatively charged DNA is wrapped around the positively charged histone octamer, a unit of 8 histone molecule, and this forms the nucleosomes. Histones are positively charged protein that are rich in basic amino acid, arginine and lysine. It is important. It is a very important point to be noted. Histone protein. This figure is exactly of the NCRT, and below that is the beaded structure. Okay. The DNA in the nucleoid is organized in large loops held by the proteins. Histones are organized to form a unit of 8 molecules called as histone octamer. And this in the length of E. coli DNA is 1.36 millimeter. Can you calculate the number of the basic pairs of E. coli? Can you calculate it? If I ask you a question as we have completed the DNA and the histone, the net electric charge on DNA and histone is, let us check. The net electric charge on DNA is, that is negative. If a double-stranded DNA contains 20% cytosine, it will have 20% guanine in it. It is true or false? Yes, it is true because the amount of cytosine and the guanine is the question number 3. If the base of adenine constitutes 30% of an isolated DNA fragment, then what is the expected percentage of the base of the cytosine? Now over here, adenine percent. Thank you. Thanks for the genes. Thank you.